Firstly, we know that glycolysis is also known as EMP pathway. The reason why it is known as EMP pathway because of the scientist name, and then where of Parnas pathway, which discovered this glycolysis pathway. Glycolysis is also known as universal process occurring cytoplasm. This is the main thing. If it is occurring cytoplasm, so all the cellular organisms must exhibit this pathway. So all cellular organisms exhibit glycolysis. After that, glycolysis involves glucose breakdown into two molecules of pyruvate, forming two molecules of ATP and two molecules of NADH. And it is divided into two phases, preparatory phase and pre-op phase. So firstly, we move towards the preparatory phase. In preparatory phase, we prepare glucose molecule for the formation of pyruvate molecules. So firstly, we add glucose with the molecule of ATP, and that is the activation of glucose with the help of an enzyme known as hexokinase. So glucose with the consumption of ATP forming glucose 6-phosphate, and at 6 position phosphate group will be added, and this glucose 6-phosphate after that converted into another molecule with the help of phosphoglucoisomerase known as fructose 6-phosphate, and forming fructose molecule with phosphate group at the 6th position. After that, this fructose 6-phosphate with the consumption of ATP forming fructose 1,6-bisphosphate and adding fructose at 6th position and fructose phosphate group at 6th position and 1 position. And this is the main thing. Fructose 1,6-bisphosphate is the committed step of the glycolysis. If the glucose molecule converted into fructose 1,6-bisphosphate, then it must complete the glycolysis and form the pyruvate molecule. After fructose 1,6-bisphosphate, aldolase enzyme breaks this fructose 1,6-bisphosphate into two molecules, DHEP and PDL. DHEP is dihydroxyacetone phosphate and PDL is phosphoglyceraldehyde. And these two molecules are interconvertible, like DHEP converted into PDL or PDL converted into DHEP. After that, we move with PDL, two molecules of PDL, because of this is the three carbon compound. After that, pre-op phase. In this phase, we activated the glucose molecule and break down into two molecules of PDL. After that, we form ATP and also convert it into pyruvate. So we start with PDL. So firstly, now PDL is aldehyde compound. We firstly convert it into acidic compound with the help of oxidative phosphorylation. How oxidation we add? Either we add glucose or either we remove hydrogen. So here we remove hydrogen with the help of NAD plus coenzyme. So NAD plus removes two hydrogen atoms and forming two NADH. And also we added inorganic phosphate PI molecule, not ATP. And this is the also main thing which we have to remember between PDL to 1,3-bis PDA. With the help of enzyme known as G3P dehydrogenase. After that, 1,3-bis PDA converted into 3-PDA. And this is the substrate level phosphorylation in which two ADP molecules converted into ATP molecules, and phosphate group 1,3-bis PDA converted into 3-PDA. And phosphate group removed from the first position, and only at third position there is a phosphate group. After that, this 3-PDA with the help of mutase enzyme converted into 2-PTA. And why we converted it into 2-PTA? The reason is we have to form double bond here. After that, remove this double bond. So for making the double bond, we have to remove this phosphate group from third position to second position with the help of enzyme known as mutase. After that, 2-PTA converted to phosphoenol pyruvate. Remember one thing: whenever there is an enzyme involved, there must be an indication of removal of water. So what happens here? That this OH molecule removes from there and give electrons to this molecule and form double bond. Like we have seen here, formation of double bond. So here removal of OH and formation of double bond. After that, it is formed phosphoenol pyruvate, and with the help of enzyme known as pyruvate kinase, this phosphate group removes from this position and donates their electron to this position, and this molecule now breaks double bond and forms single bond and makes one hydrogen here. So it makes CH3 here, phosphate group removed and give electron there, so makes double bond with oxygen, and 2 ATP is formed here. So there are total 10 steps, in which 7 steps are irreversible steps, 7 steps are reversible steps, 3 are irreversible steps. First step is a reversible step in which ATP is consumed. Third step is irreversible in which ATP is consumed, and last 10 step is irreversible in which ATP is produced. And remember one thing, this is a 1,3-bit PDA and 3 PDA in which ATP is forming, but it is not an irreversible state. It is a reversible state. And how much and total net reaction is glucose we utilize 2 ATP, we give 2 NAD plus, we give form 2 pyruvate molecule, total 4 ATP is formed and total 2 NADH plus H plus formed, and net ATP is 4, which in preparatory phase form 2 molecules of ATP, which in form in payoff phase, and 2 which we give in preparatory phase, and 4 minus 2, total 2 ATP is formed in glucose breakdown and 2 pyruvate molecule.